Hello survivors of Miss Risk's IB Chem class. My video project is going to talk about the vital role that hydrogen bonding plays in our everyday lives, especially in our DNA. So the first kind of thing to kind of know is what DNA is. So most of you probably know DNA as seen in its kind of double helix structure, which is like something like that. And this double helix structure does capture the entirety of DNA, but it doesn't really give us much as to what DNA is, how it's made up, what are the stuff that makes up DNA. So the basic unit that makes up DNA, which is called the monomer, is nucleotides. And so nucleotides are actually fairly complex. They actually look something like this. They're a ring of carbons, and they look something like this. I'm just gonna draw it out. So a nucleotide is made up of kind of three main parts, and the first part is the sugar, which is kind of this part right here. And the second part of the nucleotide is made up of something called a nitrogenous base, which is this part right here. And then the last part of the nucleotide is made up of a phosphate group, which is this part up here. So all three parts combined make up one nucleotide. And so that's kind of a lot, So that's why, chemists and biologists, being lazy as they are, they made a shorthand version of writing a nucleotide, and that's kind of the version I'm going to be using. So this part, which is the sugar, is written kind of literally as a pentagon, like that. The base is written kind of literally like that, and the phosphate group is kind of written as a P with a circle over it. So this is kind of, this is much easier to write. This also represents one nucleotide. But if you wanted to look at the actual chemicals, the actual kind of bonding and elements that make up a nucleotide, you would look at this. And this base, I'll get more into that later. So this is one nucleotide. And this is kind of what makes up each one of these kind of helices, as you will, of DNA. So for these bases that I mentioned, there are basically four types of bases, and they are called adenine, thymine, cytosine, and guanine. And each of these bases are, they're four possible bases that can be put into a nucleotide. So if you kind of really want to look at it, adenine looks something like that. And so this would be adenine right there, guanine right there, cytosine right there, and thymine right there. And that's kind of how each one of those bases look like. So if you actually wanted to draw out the chemical or the full chemical makeup of a nucleotide, you would draw the sugar, the phosphate group, and then whichever base you wanted to draw, you would actually fully draw it out. So a nucleotide is fairly complex. But these nucleotides basically form and make up the double helix structure. And these nucleotides bond together to make phosphodiester bonds. So I'm going to just focus on the phosphate group of one nucleotide and the sugar part of another one. So let's just say we have a phosphate group right here. And we have an OH. And meanwhile, you have another carbon. We have another kind of nucleotide going on right here. And the B stands for base. So each one of these carbons are linked to a hydrogen and an OH. And that's kind of, the shorthand doesn't write that out. And I probably should have mentioned that here. But each of these carbons are connected to a hydrogen and an OH. So what ends up happening in a phosphodiester bond, or which are basically how nucleotides link together, is this OH and this OH bond together and they form water, which is H2O. and what's left is just an oxygen. So what that ends up looking like is you have a phosphorus linked with an oxygen, linked with the carbon, which is then linked to the rest of the nucleotide. So, and this phosphate, or this phosphorus, this phosphate group is linked to its own nucleotide. So this process, and this is called the condensation reaction. This is how nucleotides link it together. And these bonds that form, this entire bond right here, this is called a phosphodiester bond. And that name is really long, but it's actually really easy if you break it down. Phospho refers to the phosphate group, or the phosphorus. The di refers to 2, the number 2. And then ester refers to ester bonds, like we learned in organic chemistry. And in our OChem, in our organic chemistry, we learned ester bonds as kind of carbon linked double bond with an oxygen, which is linked to an oxygen, which is linked to kind of the rest of the molecule. Well, in biology, this element right here doesn't necessarily have to be a carbon. It can be phosphorus, which is what it is in this case. So the reason it's a diester is because there's one ester bond right there, and there's another one right there. So that's kind of where it gets its name. So those are how these nucleotides kind of link together, and that's an important thing to know. So here I've kind of drawn 2 strands of nucleotides that that would kind of interact with each other in DNA. So here's one nucleotide strand right there, and here's another one right here. And if you notice, I kind of drew the nucleotides in a shorthand version, but I drew the bases kind of in their expanded form. And the reason I did that was because I wanted to kind of show the hydrogen bonding that occurs in DNA. So this is thymine right here, and this is adenine right here. And down here we have guanine, and we have cytosine right here. So guanine pairs with cytosine, thymine pairs with adenine. These are complementary bases. So between adenine and thymine forms 2 hydrogen bonds, and that occurs between this H right here and this oxygen right there. That in and itself forms a bond. And the other bond forms between this hydrogen right here and this nitrogen right there. Now some people ask, why isn't there a form between, this kind of hydrogen and this oxygen right there? Well, the reason is because if you were to look at this in 3D geometry, like this is written in paper, but if you were to actually view it in 3D geometry, you would kind of see that this hydrogen and this oxygen are not close enough to actually form a bond. So for this reason, thymine and adenine form 2 hydrogen bonds. And this is not really that important in terms of understanding how DNA is formed, but it is important when you look at DNA replication. We're not going to look at that, so don't worry about it. So between cytosine and guanine forms 3 hydrogen bonds. And that occurs between, and cytosine and guanine actually have some sort of symmetry to them. You kind of have this double bonded oxygen on each of them, and you also have this kind of NH sticking out. And that actually, the symmetry actually allows them to have more hydrogen bonds, because this hydrogen bonds with this oxygen right here, this hydrogen bonds with this oxygen down here, and then this hydrogen bonds with this nitrogen right there. So cytosine and guanine have 3 hydrogen bonds. And if you were to kind of re-look at our double helix that we kind of drew near the beginning of this video, you would see that, this would be adenine, for example, and thymine would connect right to it. This would be cytosine, for example, and then guanine would connect right to it. So these base pairs, these pairs that we saw right here, these hydrogen bonds that occur between these bases are what is holding these DNA structures together. And to think that if we didn't have this hydrogen bonding occurring between these base pairs, our DNA would be something of a single strand. And the reason that our DNA is in the double helix structure that it is because this forms kind of a protection for these complementary base pairs. And these base pairs, the sequence of base pairs makes up our DNA. For example, adenine, adenine, guanine, cytosine. This makes up a specific trait of our DNA. And just to make this clear, these nucleotide strands that we see right here are basically these lines in the DNA. Whenever we draw a double helix, these outer lines are basically the string of nucleotides paired together. And what's happening on the inside are the bases pairing together. So these bases are holding together this kind of structure, and this double helix is in the structure that it is to protect these base pairs, to protect our genetic information. And to think that hydrogen bonding is the reason that the structure can take shape. And hydrogen bonding basically makes us who we are. It basically allows our DNA to stay intact, which allows us to be kind of different people, which allows our body to successfully code these DNA strands. And so, if you look at hydrogen bonding within DNA molecules, we can see that clearly hydrogen bonding is very, very important.